Hi everyone. In class 5, we started to look at the primary structure of proteins and polypeptides and peptides. The primary structure is basically the amino acid sequence, typically going from the N-terminus to the C-terminus. In this video, I want to go over how to determine the primary structure using enzymes, and particularly proteases, enzymes that cleave peptides at certain residues. Here on the cover we have chymotrypsin, one of the enzymes we'll be talking about, and it selectively cleaves after the phenylalanine or tyrosine or tryptophan amino acid on the carboxyl side. Okay, so the idea is we have a peptide or a sample of a peptide, and we are going to create fragments and analyze each of those fragments using maybe Edman degradation, and then piece those fragments or puzzle pieces together to get the full sequence. You may ask, well, why can't we just sequence the whole thing using Edman degradation? Well, that doesn't work with larger proteins, so we're going to first cut them up into smaller fragments using certain enzymes. Now, let me go over three enzymes, or let me list three enzymes and where they cut, and we're also going to look at one chemical that selectively cuts after the methionine residue. Okay, we have three enzymes. Carboxypeptidase clips off the last amino acid at the C-terminus, and it only clips that one amino acid off. It doesn't keep clipping, is what I mean. Chymotrypsin cuts after aromatic amino acids phenylalanine, tyrosine, and tryptophan. And those are the three aromatic amino acids that I want my class to memorize. And they cut, or chymotrypsin cuts at the C side. And I'll show you an example. What do I mean by C side versus N side? Trypsin cuts after basic amino acids, lysine and arginine, but not histidine. It won't cut after histidine, and it also cuts at the C side of each of those two amino acids. Now, there is a note for these two enzymes. Chymotrypsin and trypsin cannot cut if the amino acid proline is at the cut site. So if we have, for instance, lysine, then proline, trypsin will not be able to cut between lysine and proline. But if it was lysine and valine, then it will cut in between lysine and valine. We also have one chemical, very interesting, only 3 atoms, and it can do selective cleavage. It's cyanogen bromide, that's the structure, and it cuts on the carboxyl side, or the C side of methionine. Methionine is, if you haven't memorized your amino acids yet, methionine has a sulfur in it, but not a thiol. The sulfur is within the chain. Looks like this. 1, 2, 2 methylene groups. 1, 2, then the S, and then capped with a methyl. Very interesting that this small molecule is selected for this amino acid. Okay, let me draw out an amino acid, or sorry, a peptide sequence, and we'll identify places where each of these enzymes or a small molecule can cut. Okay, based on these rules, let's see where we could possibly cut this peptide. and carboxypeptidase cleaves off the last amino acid to the C terminus. Whenever you see a sequence written out like this, assume that, unless they explicitly say so, otherwise assume that you're going from the N terminus, we'll just write this H2N, to the C terminus, the carboxyl group. Oh, sorry, you don't see that. Okay, carboxypeptidase cleaves off the last amino acid, so we're going to write here in purple, cut, and that is carboxypeptidase. And that's it. Oh, by the way, these proteases, enzymes that cleave and cut into pieces proteins and polypeptides and peptides, they typically end in ACE, carboxypeptidase, or they are trypsin, chymotrypsin, in this case just plain trypsin. Okay, that's carboxypeptidase. Chymotrypsin, we have in green, cuts after the aromatic amino acids, the ones that have benzene rings. At the C side, okay, here is phenylalanine, and so chymotrypsin can cut here. Okay, let's keep walking. Tyrosine cuts here, and tryptophan cuts here. Okay. Oh, we have to remember that chymotrypsin cannot cleave if the amino acid proline is at the cut site. I think we actually saw that, unfortunately, right here. So we can't, or chymotrypsin can't cut here. Let's put here, and can't cut. It has something to do with proline being it was that odd amino acid that where the ring is involving the amino group. So we're forming a tertiary amide right here, and also prolines typically bend the peptide in a weird manner that these other amide bonds do not. Like all of these other amide bonds are the standard amide bonds that look like this. Let's see, that's the carboxylin, right? NH, and you have your R groups like this. Okay, but proline's weird because its amide bond is actually involved, or sorry, not involved, but it's part of the R group. So you have N and how's So your R group is actually tied to that nitrogen. So that you can imagine the different geometries you can have between amide bonds that look like this and an amide bond that looks like that. This is the proline. So here's your carboxylin for proline. Okay. Trypsin cuts after basic amino acids, lysine and arginine, but not histidine. So here's trypsin. And lysine, and here's arginine. Okay, these two cuts. Okay, and then the last thing is a cyanogen bromide, and it's after methionine. So you have a met here, and how should I write this? Sometimes it's just shorthanded as the condensed structure, and it's going to cut here. It's always at the C side here, and for all of these enzymes and chemicals, and here and there. Okay, So what's the process? The process is you take maybe 4 aliquots of your polypeptide and you treat 1 aliquot with carboxypeptidase. And you'll have fragments. You'll have, in this case, a valine single amino acid, and you'll have this long peptide. Okay. So that's the analysis for carboxypeptidase, and you have those 2 pieces. Chymotrypsin, you give the enzyme enough time and it will cut in 2 places, here and here, giving us, I think, 3 pieces, right? This piece in the middle, and again, valine. So you have those fragments. Trypsin would cut twice in this case, so you'll have 1 fragment of a dipeptide, you'll have this fragment, and then you'll have a tripeptide fragment here. Again, same thing with cyanogen bromide, and it will cut twice, and you'll have 3 pieces as well, but different fragments. And by analyzing the types of fragments you have using 1 or more of these cleaving reactions, one can piece together typically the whole amino acid sequence. Okay. So memorize where they cut. Okay. It's very interesting when you memorize the structure of the amino acids. Again, you have 3 aromatic amino acids. You actually have 3 basic amino acids, but trypsin only works with lysine and arginine. You're going to put all of that together and solve a problem like this. I won't solve it here for you on this video, but look at the data that this group was able to get, and one should be able to piece together the primary structure or the amino acid sequence using these observations. So they did a full digest, they hydrolyzed every amide bond, and they quantified each amino acid, and they realized that they have a peptide hormone that has these 8 amino acids, and it's used once. Each of these amino acids is used once in the peptide. They did the following 3 experiments. They did a trypsin cleavage, cyanogen bromide cleavage, and a chymotrypsin cleavage. Okay? What they're not telling you is, after they do the cleavage, they're not telling you the order. So they're saying here, they did a trypsin cleavage and they cut once. There's a hexapeptide and there's a dipeptide, but you can't assume that this is N to C. This is not connection. This is a single amino acid, aspartic acid, and a single amino acid, lysine. That's in the dipeptide, but you don't know the order. But you could figure out the order using what we learned here. It's a lot of trial and error. It's a puzzle, basically. Same thing here, right? You have 2 tetrapeptides fragments, but they don't know the order of the 4 amino acids. They just know that they have 2 tetrapeptides. They want to know what is the primary structure of the peptide. So go ahead and take each of these clues, make some guesses. Hopefully you can eliminate all your guesses until you get to one structure that is consistent with all of these observations.